and for the opportunity. I'm going to give a perspective on RNA persistence and recovery from acute RNA virus infections. And first, some examples of studies that we've done, and then how it may relate and likely relates, we think, to long COVID and the complications after SARS-CoV-2 infection. So we've studied the first observation that RNA was not cleared after what was apparently acute virus infection was in studying encephalitis with alpha viruses, where the target cells are neurons. And so these are long-lived cells that obviously the host can afford to do without. So recovery from virus infection of these types of cells, here seen motor neurons infected with alpha virus, require the immune system to have a non-cytolytic approach to controlling virus infections. And we found that in the red here is the period of time during which you can recover infectious virus. So basically this infection is cleared very rapidly from the brain of, these are mice that are infected with this virus. However, if you look at the RNA for the virus, you find that it's cleared very slowly and actually is not completely cleared, so that it's present for the life of the animal. Now, we looked at infectious virus. We identified these non-cytolytic approaches that the immune system uses to clear infection, so antibody and interferon gamma. And so the infected neurons survive from infection, and then are the source then of this RNA that's persistent for long periods of time. Well, a consequence of that, if you look overall, you see infection of the neurons with infectious virus being produced, but that gets shut off fairly fast with the innate response, plus T cells that are making interferon gamma and B cells that are making antibody that infiltrate the nervous system to control and shut down production of infectious virus, but do not kill the neuron and allow the neuron to remain, but we have persistent viral RNA. So that means that there's actually a requirement or a need for long-term control. And so what you also find in the brains of these mice is then long-term residence of both B cells making antibody and T cells making interferon gamma that we assume are preventing the reactivation of this infection and progressive disease. Now, that was all with neurons, and neurons are long-lived cells, as I say, and essential and not replaceable, not easily replaceable, that's for sure. So we were very surprised to, and did not think that this would be true of other acute virus infections. So this is the case of measles, which I think is most relevant to thinking about SARS-CoV-2 and long COVID. So in this case, these sites of virus replication are in lymphocytes and epithelial cells, all cells that can be replaced. They're not neurons. And so the blue is the period of time during which you can find infectious virus after infection. It's a respiratory infection that becomes systemic. The onset of the rash is the adaptive immune response. You see a clearance, and you could no longer recover infectious virus after about 2 weeks. But that's not true for RNA. You could continue to find viral RNA in peripheral blood mononuclear cells for months after apparent recovery from disease. But if you look in lymph nodes, you can find viral RNA there for many more, long after it's apparently cleared from the blood and from peripheral blood mononuclear cells. And we don't actually know how long it takes to clear that infection. But what's relevant for our current discussion is the fact that if you look during this period of time at what's going on with the immune response after apparent recovery from the acute infection, there's a lot going on. So early on, you find interferon gamma T cells. Later, you find these IL-17 positive T cells. You see increasing appearance of cells that are important for making antibody. You see maturation of antibody. So this is a very active immunologic response to infection that's occurring, as I say, months after apparent recovery from the infection. And for measles, it's also relevant that it's an immunosuppressive disease. And so this persistence of RNA may well be very important for the alterations of immune responses that are seen for, again, years after recovery from measles. So I'd just like to point out that even though we think of virus infections killing the cells if the immune response doesn't, actually in vivo, it's very common for cells to survive. And there are a lot of mechanisms that are both the viral mechanisms for establishing persistence and for preventing cell death, as well as host mechanisms. Non-cytokine clearance we've already mentioned with respect to alpha viruses, but the sites of virus replication often are infiltrated with immunosuppressive cells and responses. And the virus has many ways to dampen the amount of virus that's being made and the replication in order, we think, to actually avoid killing this infected cell. So there's lots of examples of acute RNA virus infections where there are late complications. For measles, subacute sclerosing panencephalitis, which is a nervous system disease, occurs 8 or 10 years later. We think that RNA is driving the development of lifelong protective immunity. We're in the process of looking at that. But I'll just point out that for Ebola virus, there's late transmission occurs, recurrent uveitis, encephalitis. There's a post-viral syndrome that is very profound after people have, as I say, apparently recovered from the acute infection, cardiomyopathy with enteroviruses, and then the long COVID of SARS-CoV-2. So what evidence do we have that persistent RNA may be driving some of the complications that are seen or the symptoms that are seen? Well, there's certainly detection of RNA in a number of sites. Respiratory secretions and stool are the most commonly examined, and where that's been found. And certainly there's one study at least that reports RNA and protein in a subset of blood monocytes that also is very prolonged. And autopsy studies have shown widespread detection of RNA in many tissues. Of course, those are people who died rather than long COVID, but it raises the possibility that this has happened in more commonly. And so we're focusing right now on seeing if there is any evidence for prolonged virus-specific immune activation that occurs in association with people who have long COVID versus those who don't, basically to see if you could get evidence that RNA is driving any of these symptoms. So that's a summary of, I think that's the last slide, yes. So in conclusion, basically, persistence of viral RNA after acute infections is common. I personally think that it's the rule that it usually occurs. Both viral and host mechanisms foster suppression of production of infectious virus and survival of infected cells. And there are consequences that are varied and host and virus dependent, depends on where this is happening, the characteristics of the virus itself. But we know that cellular dysfunction can be progressive persistent cytokine chemokine production as possible, long-term recurrent stimulation of adaptive immune responses, late reactivation of infectious virus production, and unexpected transmission are all potential consequences. So I think that it certainly is conceptually possible that long COVID is being driven, although with the various pathogenic mechanisms may be related to the persistence of viral RNA. Thank you. Quit sharing here. Okay.
All right. Thank you so much, Dr. Griffin. We will now be moving to